Hi.
Yes,
My
yes, we can hear you.
okay. Let's get start my presentation. And my name is Takayuki Yoshimoto. I am basic immunologist studying the regulation of regeneration and immunity and anti-tumor response in the field of regenerative medicine and oncology and immunology and so on. So today I'm going to briefly talk about characterization of Kintaro cells, mesenchymal stem cell, and also briefly introduce the relationship between mesenchymal stem cells and infectious disease using a mouse influenza virus infection. And the Kintaro cells, as already talked by the previous speakers, Kintaro cells have very great clinical success in many Southeast Asian. And Kintaro Cells Powers Corporation have been doing a collaborative study with our Tokyo Medical University for more than 3 years ago. First of all, with Dr. Oyashiki, who is a former professor of Department of Hematology, and then I have taken over this collaborative study from last year. And then our objective of our collaborative study is to obtain proof of concept of application of Kintaro cells to human patients in Japan. So we are currently doing some kind of basic research to enhance the therapeutic potential of MSC. So this is also, as talked by the previous speakers, mesenchymal stem cell-based therapy holds most promising potential in regenerative medicine. And currently, the highest number of clinical trials for these autoimmune inflammatory diseases are currently ongoing. But this is a summary of characterization of mesenchymal stem cell. But these diseases are kind of autoimmune disease and inflammatory disease. And today, from the point of view of immunology, in general speaking, disease can be mainly classified into two categories. One is autoimmune disease and inflammatory disease and allergic disease, and another is a cancer and infectious disease. So autoimmune disease is a kind of a chronic inflammation, and allergic disease is a hypersensitive immune response, and inflammatory disease is an excessive immune response. But in contrast, cancer is a kind of cells escaped from immunosurveillance, and for infectious disease, host defense, and against pathogen is necessary. So different. So for this kind of autoimmune disease and inflammatory disease, as a therapeutic strategy, for this disease, inhibit immune response and the inflammatory response is necessary. So mesenchymal stem cells very good at induce such kind of inhibitory response. But in contrast, for this cancer and inflammatory disease, infectious disease, augmentation of immune response and inflammatory response are necessary. But the MSC is not so good at induce such kind of augmentation of immune response. But today's talk about this cancer and infectious disease. So to understand a little bit complicated story, there is a nice concept that mesenchymal stem cell is recently published in paper. There is a concept that the mesenchymal stem cell can be polarized into a pro-inflammatory state called MSC1 and also anti-inflammatory state called MSC2, depending on the inflammatory environment, environmental stimulation. So this concept is kind of MSC function as a sensor or switcher of inflammation. So during early phase of infection, in response to the pathogen, MSC is polarized into the pro-inflammatory MSC1 through the toll-like receptor 4 receptor to augment the activation T cell and host defense against the pathogen and anti-tumor effect. In contrast, during late phase of infection, in response to the dumps such as DNA, dead cell-derived DNA and RNA through toll-like receptor 3, MSC differential polarized into the anti-pro-inflammatory MSC2 phenotype and augment the induction of regulatory T cell and tissue generation and the pro-inflammatory effect. So So today's talk, first of all, I would like to talk about the effect of Kintaro cells on mesenchymal, effect of tumor growth and angiogenesis, and also characterization of Kintaro cells bone marrow MSC. These data was recently obtained by Dr. Oyashi, previous Professor Oyashi's group. And then, these data are currently being published, submitted to the international journal and accepted, and is currently in press. And first of all, here we compare that young and old donor-derived bone marrow MSC and characterized as shown here. First of all, these two photographs is a morphological appearance in phase contrast microscopy, and this is a young bone marrow MSC-derived MSC. This is an old donor-derived bone marrow MSC. Actually, apparent the phenotype is very different. And typically, fibroblast-like spindle-shaped morphology both. And then cell surface analysis, cell surface anti-analysis are performed as shown here by FACS analysis. As shown here, both young and old bone marrow MSC show typical MSC phenotypes, as already talked by previous speaker, that they show the typical marker like CD73 positive and CD90 positive and CD105 positive. These are all three markers for typical MSC, but negative for CD34. This is a marker for hematopoietic stem cell, and CD47, this is a marker for hematopoietic cell, and negative for MSC class 2, HLDR. And then spectral karyotype analysis are performed as shown here. Both show a normal karyotypes and no chromosomal aberration were observed in both young and old bone marrow derived MSC. And then in vitro, then their ability to differentiate into the adipocyte and osteocytes are compared. Under the adipocyte differentiation condition, young and old bone marrow MSC under these differentiation condition, differentiated to the adipocytes, which are determined by oil red or staining, and typically they show lipid droplet inside. And similarly, young and under the osteocyte differential condition shown here, and finally detected by alizarin dead staining, these young and old cells can differentiate osteocyte very similar efficiently. So these just suggest that Kintaro cells, including both and the young bone marrow MSC, have a typical normal MSC phenotype. Then we compare their effect on the tumor growth and the angiogenesis. And here, here we use the condition medium from young and bone marrow MSC and compare the old effect of old bone marrow MSC. As shown here, we use the 3 different types of cell lines, and each of which contains 3 different cell lines. So lymphoma, the multiple melanoma, and leukemia. And vertical axis shows cell growth determined by the live cell imaging analysis, and horizontal axis shows the time after the simulation. And red line shows the young bone marrow MSC derived condition medium, and blue line the old bone marrow MSC derived condition medium. And the green line is the control medium. And as shown here, for lymphoma, multiple melanoma cell line, or 6 cell lines, young bone marrow cell greatly inhibit tumor growth. Condition medium greatly inhibit tumor growth, as shown here. But old bone marrow MSC derived condition medium didn't affect at all. In contrast, for the leukemia, both young and old bone marrow MSC are rather relatively enhance the tumor growth. So these results suggest that the young bone marrow MSC, but all MSC have potent inhibitory effect on tumor growth, shown here, but a little bit depending on the type of tumor. And you know, condition medium containing not only solar factor, but also extracellular vesicle called exosome. Then we compare the effect of extravesical on the tumor growth, as shown here. As shown here, there is no effect on the tumor growth of extravesical. So that means that this inhibitory effect is due to the kind of soluble factor, kind of cytokine or something like that. And then we compare the oh, sorry. We compare the angiogenesis activity. Here, and this angiogenic activity is determined by a kind of a tube formation of HUVEC, human umbilical vein endothelial cells induced by conditioned medium obtained from 3 different types of cell lines, as shown here: lymphoma and melanoma, leukemia. As shown in the left slide, left panel, control medium greatly induced tube formation, as shown here. But the addition of condition medium of bone marrow MSC greatly inhibit all tube formation induced by lymphoma, melanoma, leukemia. But all bone marrow derived MSC condition medium didn't affect at all. And extravesical, in this case, extravesical also shows some effects, especially derived from young bone marrow MSC. So statistically calculated, as shown here, again, young bone marrow derived MMC condition is greatly inhibited lymphoma, multiple myeloma, leukemia, derived tube formation, angiogenesis, but old one not affects so much. And extravesical vesicle, like including exosome, also shows some inhibitory effect. So taken together here, taken together, young bone marrow MAC, but not all MAC have potent anti-tumor activity through inhibitory effect on tumor growth and tumor-induced angiogenesis. So now I want to switch the topic onto the effect of MSC on the immune response against virus infection. Actually, this is a little bit complicated also. Differential effect of MSC are also reported, as shown here. It is widely known that MSC can suppress T cell function, but whether MSC can also inhibit pathogen-specific T cell function remains controversial, because as shown here, actually there is an opposite report already reported. For these reports, MAC doesn't have or no effect on the virus-specific T cell induction, even in preclinical trial and also clinical trial. But according to these papers, MAC reported to show a suppressive effect for the induction of virus-specific T cell induction. So a little bit controversial. But there is a nice paper recently published by other groups here. They show that treatment MSC is very, how can I say, effective against the virus, influenza virus infection. Here they use the mouse influenza virus A H5N1 virulent virus, and then after infection with this virus, mice were infected with this virus, and after 5 days, mesenchymal stem cells were intravenously injected into mice, and as control, fibroblasts were severely injected. As shown here, mesenchymal stem cells greatly prolonged the survival rate, and also inhibited body weight loss due to the influenza virus infection. And then they also found that MAC can improve the alveolar fluid clearance and also fluid leakage into the alveolar spaces, as here. And then they also do some research on the mechanism, and they found that MAC increased the alveolar macrophage type M2 phenotype. This is a kind of inducible tissue protective phenotype in bile fluid. And then also examine the molecular mechanism. Here also they examine the lung history, Shown here, lung histopathology was also improved by the virus in mice treated with MSC. But during middle or early phase of infection, the effect of MSC is not so much. But during late phase of infection, liver injury, lung damage can be very clearly attenuated due to the treatment of MSC. Then they examined the molecular mechanism to induce such kind of protective effects. They found that enhanced angiopoietin-1 protein, this is a kind of a growth factor for endothelial cells, and also keratinocyte growth factor, KG5, secretion were observed by MSC. MSC is co-cultured with infected alveolar epithelial cell, as shown here. So they are thinking that these molecules contributed to the induction of protective effects. And then finally, they examined the lung virus titer in vivo. But as shown here, there is no difference between the lung virus titer. I mean, remain comparable. That means that there is actually no effect on the inhibition of the virus progression itself. But totally, as shown before, right, mesenchymal can reduce lung injury by improving the endothelial cells and epithelial cells barrier through production of angiopoietin-1 production in this case, and also improve the alveolar fluid clearance through the production of KG5, keratinocyte growth factor, and also resolving inflammation through the production of IL-10 or something else. So anyway, mesenchymal cell very protective against even an influenza virus infection, although their effect on inhibition of T cell specific T cell, I mean, virus itself is, I mean, similar. I mean, no effect. So eventually, interplay between MSC and the virus is kind of a, how can I say, double-edged sword, which may provide beneficial effect on lung injury, but suppressing anti-virus CTL induction. So in addition to GVHD prevention, MSC may be promising a tool for treatment of virus-associated disease such as immunological abnormality in HIV, chronic hepatitis in hepatitis B virus, and acute lung injury induced by influenza virus. So MSC may have a dual effect of like a double-edged sword, but overall, still I think, according to the paper, they show the protective. But a better understanding of the interplay between MSC and the virus is necessary to apparently delineate the safety and efficacy of mice of MSC as a cell transfer therapy. So that's it.